Everybody. I hope you're doing well during these trying times. My name is Olga Kryshtaliova, and today I am going to review the article called Little Plant, Big City, a test of adaptation to urban environments in common ragweed. The article was written by Amanda J. Gordon, David A. Mueller, and Peter Tiffin in June 27th of 2018 by Royal Society Publishing. So the article explores how urban and rural populations of ragweed have diverged in flowering time, which is a trait that strongly affects fitness. The primary objection of the study was to advance the understanding of local adaptations to urban environments in plants using realistic field experiments. Specific objections were to characterize whether urban and rural populations differ in fitness and phenotypic traits that are probably subject to strong selection, compare the extent of interpopulation phenotypic divergence among urban and rural populations, determine whether selection acting on these traits differs between urban and rural environments or between urban and rural genotypes, and test for evidence of local adaptation to urban and rural environments. By comparing the extent phenotypic diversity among urban and rural populations, scientists wanted to gain insight to how broadly environments may affect evolution. So, since urbanization causes noticeable changes in the biotic and abiotic environment, meaning urban areas are warmer, have increased levels of CO2 and ozone, greater salinity stress, and altered precipitation patterns compared to non-urban areas. These environmental changes can produce biodiversity, modify plant growth and physiology, and change animal behaviors. The evolutionary consequences of urbanizations have been studied far less than the ecological consequences. However, they are more likely to alter both non-adaptive and adaptive evolutionary processes. Urban populations are often smaller and more fragmented than rural populations, therefore increasing genetic drift and genetic differentiation among populations. Environmental differences between urban and rural environments may cause natural selection and diversity among populations. To conduct a comprehensive study of local adaptation to urban environments, these three scientists used 2 rural and 2 urban sites to transplant ragweed. In 2014, they collected seeds from multiple urban and rural populations of common ragweed in the Minneapolis-St. Paul, Minnesota area. The urban populations were collected in the city center core of Minneapolis, which has high concentration of buildings and artificial structures. The urban populations were collected from a range of habitats, including bike paths, road medians, abandoned lots, and riverbanks. The rural populations were collected from the edges of agricultural fields and roadside ditches that were outside of the urban heat island. In 2015, they planted 4 common gardens, 2 in urban areas and 2 in rural areas. At each site, they transplanted approximately 4 seedlings. Scientists visited each of these sites twice weekly to collect data on 3 phenological traits: date of transition to reproductive phase, date of first male flower, and the date of the first female flower. The transition to reproductive phase was recorded as the date on which a reproductive bud in the apical meristem first appeared. The first male flower was recorded as the date on which the first anther opened and shed pollen, and the first female flower was recorded as the date at which stigmas first appeared. Scientists also calculated the time between the first open male flower and the first open female flower. To test the fitness differences among urban populations and rural populations, they conducted two series of analyses. One analysis was conducted among data only for urban populations, and the second one only for rural populations. The results of this study seem to be consistent with the past adaptation studies. Selection tended to favor larger plants, earlier transition to reproduction, earlier flowering, and delayed time between first open male and female flower. Scientists found that plants grown in the urban sites flowered later than plants in rural areas, contradicting to natural order. In finding of earlier flowering in rural areas compared to the sites in urban areas could be related to different growth conditions, whether in weather during the year that they have conducted this experiment or just conditions at each of these field sites. Another difference occurred within urban sites. There were significant changes to male and female fitness. However, there were no such changes observed in the rural sites. The urban populations appeared to display a greater variance in both phenological and fitness traits. However, the scientists have admitted that with only 8 populations per region, the sample size was simply not large enough to detect statistical variances. So they recommend that future work should consider including both multi-year experiments and fine-scale sampling to explicitly incorporate microenvironmental variation among sites and genotypes in cities. I would like to point out that all of the works cited were from the article called Little Plant, Big City, a test of adaptation to herbal environments in common ragweeds, written by Amanda Gordon, David Mueller, and Peter Tiffin, and published in 2018 by Royal Society Publishing.